I'm Sarah Sodi with David Eichenbaum, who practices in Tampa, Florida with the Morsani Retina Group. And we're here today to talk about something very new, something that is maybe not currently being spoke about enough, which is Sozinibercept. So Dr. Eichenbaum, I'll let you take it off with just giving a brief summary, actually.
Thank you for interviewing me. Thank you for the invitation. I'm very grateful. Sozinibercept is an interesting molecule because it's a trap that inhibits VEGF-C and D. Our current commercially available anti-angiogenics inhibit VEGF-A, and there's a compensatory increase in C and D. So the thinking is if we can suppress C and D with this additional protein, we might be able to improve the efficacy of our treatment for neovascular macular degeneration, combining suppression of VEGF-A, which we're very good at, with suppression of VEGF-C and D.
Yeah, you mentioned in the talk as well about how if we're already kind of tackling VEGF-A, then maybe VEGF-C and D is subsequently just increasing further. Is that something that they have seen in the kind of the earlier phase trials?
Yeah, this is something that we've seen bear out in the phase 2B clinical trial, which is a big phase 2B trial. 366 patients in the phase 2B trial who were treated with ranibizumab, potent anti-VEGF-A suppression monthly, like the gold standard of VEGF-A suppression, treated with Sozinibercept low dose and Sozinibercept high dose in combination with ranibizumab. So when the patients were treated with ranibizumab and Sozinibercept high dose, there was an improvement in visual acuity by over 3 letters in all comers and over 5 letters in patients who were pre-specified to be analyzed without RAP and with minimally occult or minimally classic and occult lesions. So patients with these specific lesion types had over a line of superior visual improvement than with ranibizumab treatment alone, which is pretty remarkable because we haven't moved the needle on efficacy since we started treating patients with VEGF-A suppression.
Yeah, you mentioned with the phase 3 trials as well that it's not just going to be non-inferiority, that you're actually looking for those letter gains. Can you talk a little bit about how that is actually completely different than what we've been seeing previously?
Yeah, what we've been doing for approval of drugs is looking for non-inferiority in visual acuity for the most part with wet macular degeneration. In this series, we're looking for statistically significant superiority of visual acuity change over time. At the primary endpoint, it'll be 12 months. And if the Sozinibercept, which is being dosed with both ranibizumab in one phase 3 trial called SHORE and with aflibercept in another phase 3 trial called COAST, which are otherwise identical trials, what we'll see for the trials to read out positively is a statistically significant increase in vision when the Sozinibercept is administered with ranibizumab or aflibercept. How much vision is important? Well, you can get statistical significance with just a couple of letters. I'd like to see at least 4 or 5 letters of superiority to make the drug clinically significant.
I remember you mentioned even, and you were asking the audience as well for kind of how many letters would be significant for them. Is 5 that kind of cutoff that you're hearing more and more?
I think we want to hear a line of visual acuity. I think that a line would mean something to a lot of docs. I think 2 or 3 letters may not mean so much to a lot of docs to subject patients to a whole bunch of more injections. But I think if we get 5 or 6 letters, it would be almost a no-brainer, like a brainless adoption. We should do this to get our patients another line for treatment-naive patients. But that's really what we want to see. That's where the rubber meets the road, is how big is the statistical significance going to be. If it's 4 or 5 letters, then we gotta have to think about it and see more than 5 letters, we've really got this. 6 or 7 letters, we're like, over the moon of them.
Yeah, exactly. Yeah. And so I think, because we're talking more about, there is a lot of injection load. So now, even within an office, you maybe are injecting GA patients, then you have IMD. How does this kind of add to it? Is this something where you think over time, though, the injection load will decrease?
Yeah.
How do you kind of manage?
An excellent question. In the trial, they're injected on the same day. In the real world, we may or may not do that, depending on flow, patient preference, access, reimbursement, all the pressures of real world versus clinical trial environment. How long would you have to treat patients? Let's assume it works. Let's assume it gives you a line. How long would you treat patients in combination? Well, in the study, they're being treated for 100 weeks, primary endpoint of 52 weeks. We kind of have to see what those improvement curves look like. Based on the MOA and the clinical science and my understanding of the disease, I don't think it would be forever. We have to see what the right period of time would be. We also have to see if it would benefit treatment-experienced patients. That's something that has to be studied separately outside of this clinical trial and clinical development program.
Yeah. And so you mentioned that COAST has already finished enrollment. SHORE is a couple of weeks out. When do you think we'll get the first kind of look into these results?
So I would say, assuming that SHORE finishes enrollment this quarter, as it's predicted, I think that it's likely that both studies would probably release results together because they're ending enrollment close to the same time. So I would say probably in about 14 to 16 months, we'd see top line results.
Fantastic. Well, we look forward to seeing those results. Thank you so
for having
much
me.
for
I
having
appreciate it.
and for giving us a great presentation and a great talk. And we'll see you soon.
See you soon.
clinicianlink.ai